Hi everyone, and welcome to this series of videos intended to help you better understand content as you work through the Kaplan MCAT books. We'll be going through content chapter by chapter, and we'll tackle a few practice questions along the way. Today we'll be looking at chapter 1 of the biochemistry section, amino acids, peptides, and proteins. Let's get into it. Let's start off by talking about amino acids. What are they? Amino acids are the molecules that make up proteins. They contain two main functional groups, an amino group, as we can see right over here, and a carboxyl group, as we can see over here. The central carbon is known as the alpha carbon, and both functional groups will be attached to this alpha carbon. Also attached to the alpha carbon is a hydrogen atom and an R group. This R group is also known as the side chain, and this is unique to each amino acid. It will be important in determining its properties, as we will soon see. So we'll be discussing stereochemistry in another section, but it is important to note that with the exception of glycine, that has two hydrogens as we're soon going to see, all the amino acids have a chiral center, making them optically active. Glycine is achiral and hence not optically active. Amino acids can be L amino acids, which correspond to an S configuration, or they can be D amino acids, which correspond to an R configuration. In eukaryotes, the chiral amino acids are L amino acids. So a common question type that you'll be seeing in practice is the question that we have above. So let's kind of work through that and see where we get. So of these two amino acids, which one would be found in a mammal? So first off, looking at this question, we know that a mammal is a type of eukaryote, and we just talked about the fact that in eukaryotes, we are only going to be seeing L-type amino acids. So the goal of this question is to get us to determine which of the two amino acids below is an L-type amino acid. Now keep in mind that on the real MCAT, you're going to have very few of these discrete questions. They're mostly going to be hidden in passages, but it is important to understand that baseline knowledge to kind of work through those passage questions. So the first step, and just make sure that you have a good understanding of stereochemistry. If not, you can refer to the organic chemistry chapter 2 for a more detailed explanation on RS isomers. But we are going to start off by assigning priority. So after assigning priority, we can see that on the left-hand side, we have a counterclockwise movement if our three priority groups, so I know here I have a clockwise arrow, but keep in mind that when we assign priority and determine R or S, we want to make sure that our priority group number 1, 2, and 3 are facing the front. So when they're facing the front, we actually have an S configuration. And then moving on to our amino acid on the right-hand side, we have a R configuration, as we can see. So if the three priority groups were facing the front, we would actually be moving in a clockwise fashion. So with that knowledge, we know that an S configuration corresponds with an L amino acid and an R configuration corresponds with the D amino acid. And we also know that in a eukaryote, we are only going to be finding L amino acids. So therefore, the amino acid on the left is the one that we are going to be seeing in a mammal. Let's also take a quick look at cysteine. So cysteine is a bit different. In eukaryotes, again, we will only have this as an L amino acid, but it will actually have an R configuration. This is the case because here we have the thiol group being assigned priority before the carboxyl group here is assigned priority. So in this case, we are actually moving from group 1 to group 2 to group 3. We are moving in a clockwise direction, We're moving in a clockwise direction, meaning that it has S configuration. So just to note, the reason the thiol group has priority over the carboxyl group is because sulfur is greater in terms of atomic number than oxygen is. And if we were to begin looking at this to assign priority, we start off with carbons on both ends. So we have a carbon here. And as we know, we're going to move on to the next atom that is attached to this carbon. So in this case, we have that sulfur, and in this case, we have oxygen. And as we just saw in the periodic table, sulfur does have a greater atomic number and therefore will have higher priority. So we will be moving this way, and it's an R configuration that we're looking at here. Now let's talk about the amino acids you'll need to know on the MCAT. There are 20 alpha amino acids that are encoded for, and these are known as proteinogenic amino acids. I like to break down amino acids into nonpolar, polar, and charged categories to better remember them. Let's first look at nonpolar amino acids. That's what we're looking at over here. I like to use mnemonics for all my studying. I found it to be very helpful. And the mnemonic I like to use to remember these nonpolar amino acids is Vector and I love playing games while making food. So the first letter of each of these words corresponds to the one-letter code of the amino acids. On the MCAT, you are expected to know the structure of each amino acid, the three-letter code, the one-letter code, and obviously the full name of the 20 proteinogenic amino acids. As the name suggests, these nonpolar amino acids have nonpolar R groups. This makes them hydrophobic in nature. This means that they are more likely to be found on the inside of proteins away from the aqueous environment at the surface. So let's just quickly walk through some special notes to remember the amino acids. So we have valine, alanine, isoleucine, and leucine, which have alkyl side groups. So these just have different strands of carbons, and there are different ways to remember them. I like to remember that valine kind of forms a V, similar to its one-letter code. And then you'll notice that leucine and isoleucine are quite similar. It's just the placement of this methyl group that's different. And alanine is just a single carbon chain. And then we're looking at methionine. So methionine has a sulfur in it, but it is nonpolar because the sulfur has a methyl group attached to it. We also see proline, which forms a ring structure. It's a cyclic amino acid. We're going to see later that this places limitations on where proline can appear in a protein, as the ring structure makes it less flexible. We're also looking at glycine. Glycine is our smallest amino acid with a hydrogen R group. So like we talked about earlier, it is achiral. Next, we have our polar amino acids, which can be remembered using the mnemonic, you seem quite terrible, no children. Of these polar amino acids, 3 of these will have hydroxyl groups, our tyrosine, our serine, and our threonine. This is important because these 3 amino acids are the only ones that can be phosphorylated due to the hydroxyl group. This is often a common question or common point that you'll have to think about in questions on the MCAT, so just keep in mind that hydroxyl groups mean that they can be phosphorylated. We also have asparagine and glutamine, and these two have amide side chains. So we will see this just on the next slide, but keep in mind that although they are similar in name and appearance, these are different from glutamate and aspartate in that they cannot gain or lose protons to become charged. And finally, we also have cysteine, which contains a thiol group, and due to the weak nature of this SH bond, it is very prone to oxidation, and we're going to see this in a quick sec. Finally, we have our charged amino acids, which along with the polar amino acids are hydrophilic and typically found facing outward at the protein surface. They can be remembered using the mnemonic, don't ever rush knitting hats. The first 2, glutamic acid and aspartic acid, are negatively charged, and the last 3, arginine, lysine, and histidine, are positively charged in physiological conditions. When we refer to these charged amino acids, it is the fact that the side chain of these amino acids, so this part over here, this nitrogen, this nitrogen, this nitrogen, these can become charged. So we're going to see this just on the next slide, but amino acids are amphoteric species, meaning that they can accept and donate protons. So in acidic conditions, when there are high amounts of hydrogen ions, our ionizable groups will gain protons. And in basic conditions, when there is low free hydrogen, they tend to become deprotonated. So let's talk about pKa. The pKa of a group is the pH at which half of the molecules are deprotonated and half are protonated. So let's kind of think about this logically. If the pH were greater than the pKa, would the majority of molecules be protonated or deprotonated? Well, if the pH was greater than the pKa, that would mean that the environment is more basic than the pH at which half the molecules are protonated. And we just talked about the fact, this is how I like to remember it, when you're in a more basic environment, there's less hydrogen floating around. So you are more prone to being deprotonated yourself, right? In a more basic environment, there's less hydrogen, and in a more basic environment, a pH that's greater than your pKa, you are more likely to be deprotonated. So when your pH is greater than your pKa, the majority of molecules will be deprotonated. On the other hand, if the pH is less than the pKa, if the environment is more acidic, if there's more free-floating hydrogen ions, you have more hydrogen to protonate the molecule. So the majority of the molecules will be protonated. Now let's take a look at glycine. All amino acids, as we've seen, have carboxyl and amino groups. Our amino group and our carboxyl group here. These two groups have the ability to be protonated and deprotonated. In this case, the amino group at the moment is deprotonated, whereas the carboxyl group is protonated. It is important to note that the pKa of this carboxyl group is about 2, whereas the pKa of this amino group is about 9. So we're going to see what this looks like in 3 different pH settings, one of those being physiological pH. In extremely acidic pHs, let's say for example a pH of 1.5, the carboxyl group here, which has a pKa of 2, we have a situation in which the pH is less than the pKa. The pKa is 2 and the pH is 1.5. As we talked about on the previous slide, this is going to mean that our carboxyl group is protonated. So here we have a charge of 0 because we have a proton right here. At the same time, our amino group, the pKa of our amino group is greater than the pH. So 1.5, pH of 1.5 is less than a pKa of 9, and our amino group will be protonated as well. So here we have a charge of +1. Overall, in these conditions, we have a net charge of +1. Now let's look at another situation at physiological pH, which is about 7.4. In this case, the pH is greater than the pKa of our carboxyl group, and this is going to mean that our carboxyl group is deprotonated. So here we have a charge of -1 on this side. At the same time, the pH is less than the pKa of our amino group. So our amino group will still be protonated. We have a charge of +1 on this side. Overall, we have a charge of 0 because we have a negative charge through our carboxyl group and a positive charge through our amino group. We end up with the zwitterion, meaning that we have both positively charged groups and negatively charged groups, leading to an overall neutral charge. Finally, let's look at an extremely basic pH, let's say 10. In this case, we end up with the pKa being greater than both the carboxyl groups and the amino groups. So both the carboxyl and amino groups will be deprotonated. In the case of the amino group, this leads to a charge of 0, whereas our carboxyl group has a charge of -1 at this point. So overall, our molecule has a charge of -1. The pI, this is another term we have here, is the pH at which the molecule is electrically neutral. There are a few formulas that are important to remember to calculate the pI. For uncharged amino acids, the pI is equal to the pKa of the amino plus the pKa of the carboxyl group divided by 2. For acidic amino acids, so this includes our glutamic acid and our aspartic acid, our pI is equal to the pKa of our side chain plus the pKa of our carboxyl group over 2. For basic amino acids like our histidine, lysine, and arginine, the pI is equal to the pKa of our R group plus the pKa of our amino group over 2. Now that we understand amino acid basics, let's focus in on how these units are going to come together to become proteins. On the previous slide right here, this is a peptide bond. So this is a covalent peptide bond. This is what's going to stabilize our primary structure. When a protein is denatured, the primary structure is typically kept intact. Next up is our secondary structure. So this is the local structure of neighboring amino acids. And we have two very common structures that are formed. We have our alpha helices, as you can see on the right, and beta sheets on the left. The alpha helix is stabilized by intramolecular hydrogen bonds. So these bonds are between the carbonyl oxygen and the amide hydrogen 4 residues down. So it is the carbonyl oxygen on residue 1 with the amide hydrogen on residue 4. The side chains are going to point away from the helix core. So this center over here is our helix core, and as you guys can see, the R groups are pointing away, they're pointing outwards. As we mentioned before, proline has a ring-like structure, which makes it quite rigid, and as a result, it's rarely found in helices because it's going to introduce a kink. Another amino acid that we will not typically see in alpha helices is glycine because it is too flexible. We saw both of these earlier, and we noted that glycine is very small, it's very flexible, it's very prone to conformational change, and we don't want to compromise our protein like that. So we're not going to see proline and we're not going to see glycine in our alpha helices. We also have beta sheets. So beta sheets can be parallel, as we can see here. They're all going N-terminus on one side, C-terminus on the other, or they can be anti-parallel. So in this case, N-terminus on one side, C-terminus, and then we have N-terminus again. In this case, our amino acid chains are going to lie alongside each other. They're going to be held by hydrogen bonds between the carbonyl oxygen and amide hydrogen of neighboring chains. Proline will not be found in the middle of these chains, again as it introduces a kink, but it can be found in turns between the chains. So we have turns here, we have a turn here. Once this is done, we can move on to the next level. This is tertiary structure. This is the protein's 3D shape and will be determined by interactions between the R groups. Hydrophobic amino acids like to stay inside the proteins, as we discussed, while hydrophilic residues are found on the surface. There are some important bonds to note, such as salt bridges, which are going to be formed between charged amino acids. So these are ionic connections. As you can see, you have a negatively charged and a positively charged amino acids coming together. We also have disulfide bonds. So we talked about how cysteine is typically oxidized due to the weak nature of that SH. So this is going to allow for this covalent bond. These are our disulfide bonds, and these can only be broken when the protein is in a reducing environment. So they are formed in oxidizing environments and they will be broken in reducing environments. So let's quickly go over why exactly the residues are located where they are. We said that hydrophobic amino acids are typically inside, whereas hydrophilic ones are found on the surface. Well, remember that entropy is the degree of disorder. Positive changes in entropy or increasing disorganization is favorable for a spontaneous reaction. If a hydrophobic residue faces the aqueous environment, water is going to start clumping together into organized clumps. This is going to lead to a decrease in entropy, which is not favorable. The hydrophilic residues being outside is going to promote that water moving around and interacting with the residues, increasing disorganization, increasing entropy, and as a result, favoring that spontaneous reaction. Finally, we reach quaternary structure. So this is only going to exist for proteins with more than one polypeptide chain. Hemoglobin is a great example of this, as it consists of 4 subunits that each bind one molecule of oxygen. This kind of structure is useful as it increases stability, reduces the amount of DNA required to encode, brings catalytic sites together, and induces cooperativity. Conjugated proteins are proteins with functional covalently attached molecules called prosthetic groups. So this can be organic molecules, minerals, ions, lipids, sugars, etc. The reverse of this folding process is known as denaturation, and this is the loss of a protein's 3D structure. Remember, as we talked about earlier, primary structure is not likely to be disrupted as a result of denaturation. Denaturation is typically caused by heat and solutes that can interfere with the kinetic energy, as heat increases kinetic energy, and this can be enough to overcome the bonds that are holding the protein together, and solutes can come in and also disrupt these bonds to break apart the three-dimensional structure of the protein. All right, so that is really it for the first chapter of... All right, so that is all for chapter 1 of biochemistry, amino acids, peptides, and proteins. Thank you all so much for watching. Please like and subscribe if you'd like to see more videos relating to MCAT studying, and if you have any feedback or suggestions for videos you'd like to see in the future, please leave a comment down below. Happy studying, and I'll see you soon.